University of Vermont, and MS from the Ohio State University. Both Heather and Rebecca will both present for about 20 minutes. We ask that everyone keep themselves on mute and hold your questions until the end. My staff's going to be watching both the chat box and those who raise their hands, and we will call upon people asking questions in the order that they were received. Again, I ask that you wait to the end to ask questions of both presentations. I also should note that we are recording this presentation, so just for everybody's reference, and we're going to share this on our SharePoint site at the end. So without further ado, I would like to hand over the presentation to Rebecca at this time, and thank you for being here.
Okay, thank you for having both of us, James. Okay, does that look all right?
Yes, it's great, Rebecca.
Okay, great. So I will be speaking today about harmful algal blooms in New York State. And just for a little orientation, we have a lot of lakes and a lot of water, and full 10% of the actual governmental state area of New York State is water. Granted, a lot of that is Lake Ontario, but we have over 16,000 small water bodies ranging from a tenth of an acre up to some of the Finger Lakes and Lake Champlain, which are quite large. In New York, we have a home rule distinction, which means there's no such thing as a private lake or a public lake. All water itself is publicly owned, but all manipulation and access to shoreline is generally related to the landowner. So the local entity has a large role on what's done on a water body. There's a large range from very oligotrophic and acidic lakes in the Adirondacks and Catskill Mountains to very eutrophic lakes and very high density water bodies, especially downstate where population density and lake density increases. There are 3 state agencies that play large roles in harmful algal bloom oversight in the state. The Department of Health has a regulatory obligation to oversee drinking water testing and works with local drinking water providers to provide those services. So to a large extent, I won't be addressing that today. They also deal with any health concerns related to exposure and symptomatic issues related to harmful algal blooms. DEC works with the DOH to provide real-time notification of bloom occurrence, and we've expanded our efforts with regards to research and mitigation. I'll be speaking about that today. And then we have the State Parks Agency as well, which all 3 entities have swimming areas that they oversee, which are beaches, and occasionally those beaches do close as a result of harmful algal blooms. And we are constantly in touch. There's a core of staff in each of the 3 agencies that oversee statewide efforts. And then, of course, there's local, county, and regional staff all over the state who are working on this topic. To a large part, all 3 agencies have agreed to take a very conservative approach when it comes to defining what an algal bloom is. Of course, there are chlorophyll, cell count, or toxin numbers to attribute to our algal blooms, but the bottom line is if there is a scum that's visually apparent, there's a potential for health effects related to either the toxins or other harmful compounds. So just the visual presence is enough to start the public notification, or in the case of a swimming beach, to close the beach. So I use this picture as an example where it's very likely that 95% of the surface area of that lake is clear of harmful algal blooms, but if that is the swimming area, that will be closed as a result of that visual scum right there. And the public communication line that we use is know it, avoid it, report it. So we really emphasize the visual recognition, physical avoidance of blooms, and then reporting it through whichever local entity is relevant. We do also have robust monitoring programs, and generally we, instead of cell counts, rely on the Flora probe as an indication of algal density. For those not familiar, the Flora probe is able to differentiate different types of algae present by their chlorophyll signatures. So it's not just chlorophyll A or phycocyanin, but all of the pigments associated with phytoplankton. And so it can give an expression of total algae and then break it down by group. And the blue-green output is mostly phycocyanin, but also other chlorophylls. Our laboratories that we work with also do a qualitative scan. So we do have a combined indication of density from the Flora probe and confirmation of cyanobacteria presence from the microscopy. And then toxins, for some measures, we rely on ELISA for the drinking water monitoring. That is all relies on ELISA. However, we also run hundreds of samples annually for additional toxins and microsystem congeners via mass spec. We have adopted since 2012 or so several different thresholds. They've changed over time as a result of new information, but the current standing is that we issue advisories, which don't really have strong regulatory impact, but we do issue notifications of high toxin presence for those two upper thresholds there. And then the bottom threshold is again, very conservative, lower than the EPA REC guidelines, but a conservative value for at which beaches can be reopened. So the bottom threshold is when a HAB has been clear for at least 24 hours, and then the beach can be reopened to swimming. And the upper two thresholds are on samples that are often collected when a visual bloom is present. So our program has moved strongly towards paired water quality sampling, where we have other indications such as nutrients and pH and temperature, in addition to harmful algal bloom parameters. And we conduct this monitoring in over 300 lakes statewide annually through a combination of our professional monitoring and a robust citizen science program in New York. We also collect visual reporting and sampling information on our notification website, which I will hopefully have time to show you today. And the full picture of harmful algal bloom sampling and reporting is conducted by a combination of professional staff, volunteers, and partner programs where we have, local park staff or academic partners. And again, all testing of drinking water through treatment trains and finished water is all under the jurisdiction of local providers and outside of what I'm presenting today. We've built together this kind of patchwork of data providers, is our internal term, over several years, and it's a combination of state agencies, federal agencies, etc. But we receive information from hundreds of lakes, and it was very important to create a sustainable and reliable way to process that information rapidly. We use our bloom status to interpret the data as it's received. So this can be based on just a photo, visual evidence, or beach closure, or it may be based on sampling information and thresholds that I mentioned earlier. The Harmful Algal Bloom Program Guide, which is on your bottom right, is a document on our website that outlines all of this in much more detail. And Diet for a Small Lake is an educational tool that our staff contributed to, which is also all available online. And then our website also has additional pages with photo galleries, a harmful algal bloom brochure, etc. We just try to put everything out there, and then local entities are able to use those resources as they wish. The most consistent outreach during the summer months is to update our statewide map of harmful algal bloom locations, and that includes beach closures, though they're not necessarily indicated as such. And we send out a weekly newsletter to over 20,000 people and have some other outreach avenues. We've been detecting and reporting harmful algal blooms since 2012, and this map indicates the extent of where they've been found all over the state. These dots represent one per water body, so some of them might be indicative of water bodies that are in bloom all summer every year, or where we've had one report once. But the variety of water bodies that harmful algal blooms are seen in never ceases to amaze me. This includes rivers, reservoirs, impoundments, large lakes, small lakes, Adirondack lakes, etc. Here's some trend data. The graph on the left shows that each year we find and document harmful algal blooms in new water bodies that were not found each year. So cumulatively, we have detected HABs in about 400 water bodies. I'll be updating this with 2020 data soon, and I expect it will rise by about the same. And once we do detect a harmful algal bloom in a water body, it does seem to recur there again. The graph on the upper right shows how many each year. So that has actually kind of leveled off. The number of unique water bodies within a year has been more stable the last 4 years. And I think 2020 will be similar. And further, the number of annual beach closures has also come down. So 2019 was lower, 2020 was lower. So there's not a clear indication that we're just seeing an astronomical rise in harmful algal blooms, and the ability to parse out how much is related to public reporting versus an actual increase in harmful algal blooms is an ever-present challenge. We've had a lot of high-profile, high media coverage harmful algal bloom events, particularly 2016, 2017, where we had some threats to drinking water in the Finger Lakes, and we have had harmful algal blooms in all 11 of the Finger Lakes, most of which are quite large, and many of which have relatively pristine, very good water quality, not water bodies where you'd expect to see harmful algal blooms, and some are quite persistent lake-wide. Further, the Wallkill River, which is a very highly modified river with a lot of dams and not very strong flow, in a drought year experienced a 30-mile long bloom, but that has not recurred there since. And also there has been several illness events. There is a paper from 2015 that summarizes a few years of illness reporting, and every year there's a few more. In 2018, particularly driven by the Finger Lakes and other media coverage, the governor's office decided to start a HABs initiative. So Governor, now many of you might have heard about Cuomo more recently in 2020, but we've been working with him for a few years. He initiated a program to focus first on 12 priority water bodies, and these were selected to cover a range of water quality and geography and harmful algal bloom history. 12 lakes, we really dug in and put out action plans, which are somewhat like a TMDL, but without a full watershed model, and really focused on why are there harmful algal blooms in this lake and what can be done about them. And there were 4 regional summits held where we gathered experts from locally and nationally to get together and discuss these 12 water bodies. And there were also planning for advanced monitoring and mitigation pilots, which I'll discuss more in a moment. And the real key effort of this initiative was to figure out why blooms are happening, how can we lessen their impact, and how can we improve water quality. So some of the research and mitigation pilots that have come out of this initiative utilize several different methodologies. Pictured here is an alum treatment on the top and a cutting edge habitats treatment, which was done in partnership with the NGOs and the Army Corps of Engineers. And we're working through the findings of these projects and have piloted several others as well. We're also working towards a research guide, which will kind of summarize the gaps in research that are needed. We are analyzing the data we have. We are conducting a statewide survey of the presence of harmful algal bloom toxins in wadable small streams, and additional mitigation pilots will be implemented over the next few years. So there's a lot going on, there's a lot coming, and we've gained a lot of staff and expertise over the last few years. The advanced monitoring pilot that I mentioned is a great partnership between a state and a federal entity. We work with the USGS Troy Water Science Center to look at a multi-dimensional study where we're looking at depth across the water surface, at the tributaries, the water quality of the tributaries coming in, and over time with a combination of grab sampling and continuous monitoring devices. And this has been really exciting. We're looking forward to evaluating the findings over the next few months. And the USGS hosts all the data for their continuous monitoring platforms on the link here. So you can go back and download data, historical data, or see it in real time. They also include webcams, so you can actually see the surface of the water, and they're really valuable. Lastly, I wanted to describe our notification system, which we implemented last summer using ArcGIS Online, and has really been a game changer in terms of processing and communicating the huge amount of information that is received daily. And the way this works is that it summarizes and interprets the information and provides internally to state agency staff certain amounts of information, and externally to the public other amounts of information. The reports are populated on a map where they remain indicated as a current HAB for 2 weeks, because we don't necessarily have follow-up data on all the HABs, so we don't indicate that a HAB has ended. We only indicate that a HAB was reported. And then users of the public can filter by their lake or their county and actually export the data themselves from the public site. The reporting is coming in using Survey 123, which is an ArcGIS Online service, and it makes it extremely easy to create high quality fillable forms that populate a cloud hosted data layer. And the forms are usable on any platform, but they're perfect for geolocating and attaching photos. So we require photos to be included on especially the forms coming in from the public, so that we can quickly evaluate whether they're looking at duckweed, water meal, or cyanobacteria. We have 2 different versions of the form, and they both feed into the same data layer. But one is for what we call trained users. That's our agency staff and our citizen scientists that are going to the same locations over and over. And so the form is pre-populated with a lot of their information on both the front end for them and the back end for us. And then the public form is a bit more questions and prompts the user to look at some photos and compare it to what they're seeing. So here's a little schematic of how it comes in. The folks report on the form. We're able to look at all of their reports and then edit them, evaluate them, and push them to the public site in a certain time frame. And then we also have a lot of results coming in from laboratories, which we, again, evaluate and decide which through to the public site. The ArcGIS Online allows you to create different interfaces to interact with the data, and it's very easy to use. So this is the 3 tiers. The top tier is our internal workspace that just the handful of core staff at the 3 primary agencies are using, and that's where we can edit the data. And the key from a source water perspective is that we can indicate on there when a water body has a public water supply, a drinking water source. So DEC can in real time say, hey, Department of Health, here's a harmful algal bloom occurring on a water body with water supply. Can you reach out to the local provider? And then they can indicate back to us, yes, we have communicated with the local provider. We don't think this poses a risk. And that seamless digital real-time communication on the same platform has been really excellent. And we have an intermediate step that is used by a lot of regional staff around the state. And then the public site does not require any login. So this has enabled us to validate and improve the quality of the data that we're maintaining. We export and use R to supplement these tools, and we have moved towards providing a memo at the start of the season to the management. Here's how we're going to prioritize our response. Here's when we're going to conduct additional sampling. And we also produce a summary each day that goes to the governor's office of harmful algal bloom reports, which is very easy to do with this system. Oops. So that's me, and I'm just going to take 1 minute to show you how they look, because we're taking it down for the season tomorrow. So normally I'd say go check it out, but it will be winterized tomorrow. So this is the public landing page that you saw a screenshot of, and our Finger Lakes are almost there in outline because they've had lake blooms all along the shore, and we have a lot of people reporting over the summer. This is that intermediate step portal, and all of these different web apps have filters, so you can say, hey, I only want the last 2 weeks. That's what's on here right now. If we turn it off, there's the whole summer. And then we could say, hey, I just want region 1, which is Long Island. So it's very quick to hop around. And then finally, oops, I can do this. Oh no. This is our workspace. So this is where we're able to go in and see a lot more of the background data and edit things as we need to, and includes a lot more information than you're seeing on the public site. So that is all from me, and I will look forward to Heather's presentation and taking questions at the end. Thank you.
Thank you, Rebecca. And Heather, I guess we'll just transfer with you.
Hi, and thanks for participating in today's webinar. Since we only have 20 minutes, I'm going to focus on some of the major HAB highlights and lessons learned, but also include a few links so you can dig a little deeper if interested. I'll start with some background. In 2010, Ohio began incident response HAB sampling at public water systems and state park beaches, and we quickly realized that cyanotoxins posed an emerging public health threat. The Ohio EPA, Department of Natural Resources, and Department of Health worked together to establish cyanotoxin thresholds for both finished drinking water and recreational waters, and created a unified state HAB response strategy. There are now separate recreational and public water system health response strategies that are typically updated annually and are available on Ohio EPA's website. Now, Rebecca focused her talk more on recreational HAB response, so I'm going to try and cover a little more on the public water system side of response. In 2013, Ohio experienced our first finished drinking water microsystems exceedance at a very small lake, Erie Public Water System, and an advisory was issued. A year later, microsystems were detected in Toledo's drinking water for multiple days. A do not drink advisory was issued, water was trucked in from neighboring areas, and the press coverage was international. Both state and federal legislators called for action. Ohio also had its first finished drinking water toxin detection, a little less well known, but it was below Ohio's drinking water threshold, thankfully, so we didn't have to issue an advisory. There's currently no national health advisory level for toxins. In 2015, Ohio EPA incorporated US EPA's new health advisory levels for microsystems and cylinders from MOPSIN into an updated response strategy and held numerous outreach and engagement sessions. We had finished water microsystem detections at 5 additional different drinking water public water systems, but thankfully repeat samples were below the newly established thresholds, and so no advisories were issued. We think this is due in part to the swift response by Ohio EPA to help water systems quickly and effectively optimize treatment and reduce finished water concentrations below thresholds. Also in 2015, Ohio Senate Bill 1 passed, which led to Ohio EPA's development of HAB monitoring and reporting rules, the first in the country. Those went into effect in 2016. The rules call for year-round routine cyanobacteria screening and microsystems monitoring at all public water systems that utilize surface water as a source. Unfortunately, I don't have time to go into all the details, but the rules and analytical methods can be found at the posted links. Ohio has continued to have limited finished water detections of microsystems and saxitoxins, and we experienced our first slinotropin and atoxin A detections in 2018. Thankfully, Ohio has not had to issue any new drinking water advisories since concentrations were either below thresholds or treatment was quickly optimized and repeat detections were avoided. The rules require cyanobacteria screening using a multiplex quantitative polymerase chain reaction, or qPCR assay. Since this is still a relatively new approach, I'll provide a few brief slides as a quick overview. For context, qPCR assays are considered the gold standard COVID testing method. The cyanobacteria qPCR assay that we use identifies and quantifies whether the genes responsible for the production of microsystems, slinotropin, and saxitoxins are present in a water sample. The assay also quantifies a gene present in all cyanobacteria, which is called 16S, which can be roughly correlated to total cyanobacteria cell counts. The assay is fast, cost-effective, uses certified reference materials, and we found it to be a more specific indicator of cyanotoxin production than traditional cell counts. The qPCR results are used by Ohio EPA to determine if monitoring for the slinotropin or saxitoxin needs to be conducted. And in addition, if systems are on reduced monitoring through our rules, they can be triggered into follow-up microsystems monitoring if the gene is detected. More information on the assay is available at the web link. We've also split samples with US EPA's research office to help evaluate and validate the method, and conducted a 4-lab round-robin comparison study. We did uncover some issues with the assay, but luckily those were quickly rectified by the manufacturer. We also incorporated additional QA in the method to help ensure differences in instrumentation did not impact results. I strongly encourage anyone that's developing a new method to do this type of rigorous evaluation when they're considering implementing new types of methods. Now we've been using this for quite a few years, so we're very happy with it. And other states are now using the approach, including Oregon, Indiana, New Jersey, probably forgetting a few. And I'll just provide a few quick examples of why we think this has been helpful. In this first example, microsystem producing genes, the MCYE, represented by blue circles, preceded the microsystem's toxin detection at this water system, shown in red, and provided an early warning to the water system. Similar trends with microsystem gene detection, sometimes preceding microsystems detections by 1 to 4 weeks, were observed at other Ohio public water systems throughout the state. In this example, the total cyanobacteria 16S data, which is represented by the gray squares, also trended well with the increase in microsystems, but the cell count data, which is shown in the purple squares, did not. While the cell count and phytoplankton enumeration helps to verify that Planktothrix was the predominant cyanobacteria in this bloom, the cell count was very low and generally would not have indicated a concern for cyanotoxin production. We found in this bloom pretty much every cell that was present seemed to be producing toxins. The drinking water source was sampled by Ohio EPA's Inland Lake program during the summers of 2014 and 2015, and microsystems were not detected, and cyanobacteria were not detected in the reservoir. It was given a clean bill of health by our Clean Water Act folks. But this example, I think, helps demonstrate the importance of this off-season monitoring. This public water system has unfortunately suffered from worsening winter blooms, and microsystems have exceeded 25 micrograms per liter in follow-up winters, and has had to upgrade their treatment to be able to address those increasing toxin challenges. This graph compares cyanobacteria cell count data, which is in the bar graphs, to saxitoxin production genes, which are the open blue circles, and saxitoxin concentrations represented by the red dots. As you can see, the saxitoxins trended very well with the saxitoxin production genes, but were not reliably predicted by the cyanobacteria cell density. And at one site, saxitoxin detections occurred with no saxitoxin producing cyanobacteria cells identified in our cell counts. We think this may be due to possible benthic production of cyanotoxins instead of cyanobacteria in the water column, and this is an emerging issue for us in Ohio. The assay was sensitive enough to detect these low gene concentrations, but the cyanobacteria cells were not present in high enough concentrations to be detected by our cell counting methods. This final example shows how the 16S total cyanobacteria genes, as in gray, compared to cyanobacteria or cyanotoxin production genes and the cyanotoxin results, lots of cyanos. The microsystem production genes, depicted again in blue, were a good predictor for microsystems, again in red, but the 16S, represented in gray, was not. The highest peak in 16S occurred in this midsummer area around August and represented a shift in the cyanobacteria community to a non-toxin producing but much higher biomass cyanobacteria bloom. So why is this important? Well, if a lake manager was relying just on cell count data alone, advisories may have been posted in midsummer when toxins were not present, but may have not been posted in early summer and late fall when microsystems were detected, cell counts were much lower. I think this is a good example of why Ohio EPA and the state relies on molecular screening methods instead of cell counts. Shifting gears, I have an entire 45-minute talk on cyanotoxin analysis methods, but I'll try and sum it up in this one slide. For microsystems, Ohio relies on microsystem ADA ELISA analysis, as multiple method comparison studies have indicated it was the best variant-independent measure of total microsystems. This is important since the health advisory level established by US EPA is for total microsystems. We do not use the US EPA LCMS-MS method because it only detects 6 of the over 200 microsystem variants and has consistently underestimated total microsystems in Ohio studies. This is especially true for our common Planktothrix blooms, which almost always produce demethylated microsystem variants and does not produce microsystem LR and is not detectable under US EPA 544 method because that method does not detect the demethylated variants. For other cyanotoxins, we do use LCMS-MS methods to complement our ELISA screening methods following any finished water detection. This is especially important for saxitoxins, since the ELISA method for saxitoxins has poor cross-reactivity for many of the saxitoxin analogs. Overall, the take-home is each cyanotoxin has its own analytical issues, and methods are not one-size-fits-all. I should also briefly mention that Ohio had some issues with US EPA's LCMS-MS anatoxin A method. We think these were due to method flexibility, but we're working through them. One of the labs participating in the UCMR4 routine monitoring had to invalidate the majority of their anatoxin A finished water detections, due in part to the data that we presented that demonstrated the lab was experiencing some false positives. It continues to be an evolving science. One thing Ohio learned through our routine monitoring was that microsystems can be present above adult health advisory concentrations in every month of the year. We found that cold-tolerant Planktothrix blooms can even occur under ice. We also found that some of our systems have their peak microsystems detection as early as May or as late as December. In a few cases, the water system did not have their first microsystem detection until November, with concentrations peaking in December and often persisting through March. Another lesson learned is HABs are not always visually apparent and can produce high toxin concentrations even when scums are not present. We often see highest concentrations during the early growth phase of a bloom. On the day that these state park photos were taken, average microsystem concentrations, and this is a ankle, knee, hip depth, 3 transects composited. Those concentrations were over 100 micrograms per liter at the beach. And I believe this is another reason for routine monitoring. Now, when routine monitoring is not possible, as the case of New York, when you have thousands and thousands of lakes, the NOAA HAB satellite products shown in the lower right can be extremely useful. This data is now available nationwide through the Federal Multi-Agency Science Project, and you can visit the provided link for more information. Ohio relies on this NOAA satellite data to develop a weekly inland lake cyanobacteria density map series. The data is used to help inform sampling decisions by local and state lake managers. In this example, a HAB was forming on the shallower north end of the lake. You can see in the kind of yellowish area, but was not present at the south beach or the water system intake. By being aware of the existing HAB, the water system could proactively optimize treatment and be prepared if a rain event could transport that HAB closer to the intake. Along with the map series, the Ohio EPA provides a weekly summary on the HAB lake status to local lake managers, water systems, and sister agencies. And I should also mention that similar to in Rebecca's talk, we do post all of our cyanotoxin monitoring results for our public water supplies on our beaches in an online portal. Another tool that is useful for HAB detection is multi-parameter sondes. Similar to, I think, the USGS study Rebecca mentioned, in one of our lakes, they found very strong correlations between microsystem concentrations and phycocyanin, which is one of the accessory pigments produced by cyanobacteria. We also found that pH is a good indicator, at least at this particular lake. And large upticks in pH have been used by many water systems as an additional alert to a potential HAB. If a lake is not dominated by cyanotoxin-producing cyanobacteria, however, the relationship between the phycocyanin, pH, and toxins would not hold. Overall, the real-time data can be useful to at least determine relative changes in water quality in your source water and the potential for cyanotoxin production. The continuous data streams from these sondes can also help fill in the gaps between your periodic graph sampling results and are also useful on lakes that are too small to detect HABs remotely via satellite. This slide shows Sourceform microsystem concentration at 4 Western Lake Erie water system intakes. What I wanted to highlight here was that rapid increases in microsystem concentrations can happen with these blooms, and increases in concentrations can impact treatment capacity. In this example, Carroll Township had a finished water detection after the concentrations in the raw water increased above 5 micrograms per liter. Carroll is an ozone plant, which is generally viewed as being highly effective in microsystems destruction, but unfortunately the ozone dose was insufficient to overcome the chemical demand associated with this additional bloom biomass, and an advisory had to be issued. They have since upgraded their ozone contactors and have not had any repeat finished water detections, even when source water concentrations were above 50 micrograms per liter. And I will say at some of our other ozone plants, our larger plants that have maybe 2-stage ozone, they can handle raw water concentrations above 100 routinely. I'll spend the next several slides covering some additional cyanotoxin treatment topics. First, to help public water systems respond to HAB, cyanotoxin treatment optimization protocol requirements were included in our rules. Cyanotoxin guidance and templates were developed to assist the water systems, and they continue to be updated based on new applied research findings. If water systems have had repeated elevated cyanotoxin detections, additional general plan requirements are triggered. The general plan is more holistic assessment of the plant's capacity, including in-plant treatment, source water protection, and reservoir management. The general plan must also include a time frame for capital improvements if existing treatment capacity is considered insufficient. I provided links to the optimization and general plan guidance, as well as a white paper that was developed in collaboration with Ohio AWWA. US EPA has also developed an excellent practical guidance on cyanotoxin treatment. Ohio's guidance is based on applied research, in many cases from studies completed at Ohio water systems. The Ohio Department of Higher Education funded 12 treatment-related applied research projects. Ohio EPA worked with US EPA on 4 HAB-focused comprehensive performance evaluations at Ohio water systems, and completed 8 powdered activated carbon jar test experiments and 2 oxidation hold time experiments at Ohio water systems. I can't thank the US EPA, ORD, and Office of Water enough for all the assistance they provided to us over the years. They've been excellent partners with both our research universities and our state agencies. In addition, treatment train data has been collected during HAB events to help assess treatment performance at the full scale. Luckily, no HAB-related drinking water advisories have occurred since the Toledo advisory. I should knock on wood when I say that, though. One thing that's kind of neat that we did was, instead of using commercial cyanotoxin standards, which can be very expensive and may not be representative of source conditions, many of the experiments I mentioned were conducted using cyanotoxins derived from the water system source water. This can be easily done by using phytoplankton net to concentrate the cyanotoxins by repeatedly pulling the net through the water column or the water surface. And we've also just put the net under a raw water tap and let the water flow through for an extended time to help concentrate the cells. We've achieved very high microsystem concentrations over 10,000 micrograms per liter using this method. You're also concentrating the biomass associated with the HAB by using this method, and that biomass can impact treatment capacity. The concentrated solution can then be spiked into raw source water to achieve a range of concentrations for controlled experiments. A benefit of this approach is you can concentrate a sample and evaluate treatment potential prior to a bloom becoming really severe. Now, in cases where a water system was already experiencing a severe bloom, just the raw water can be used in experiments. But in either case, we recommend freezing and thawing the sample 3 times to release any toxins that are inside the cyanobacteria cells if you kind of want a worst case treatment scenario. During active blooms in Ohio, we have seen extracellular microsystems concentrations of greater than 50% in source waters. This is often associated with, or we believe it's associated with cyanophages that are actively attacking cells during the active growth phase. So this is looking at 50 or more percent extracellular toxins is not unrealistic. This is just one example from an OSU study. Here we're able to demonstrate that both the type of powdered activated carbon and the amount of natural organic matter present in a sample can have a huge impact on microsystems absorption capacity. In this example, the natural organic matter used as a spike was sourced from our Ohio harmful algal bloom. We found that absorption capacity was reduced if a HAB-based NAM source was used instead of the traditional commercial standard. So not all NAM has the same effect, and commercial standards may underestimate its impact. Since powdered activated carbon is one of the most commonly used cyanotoxin treatment barriers in Ohio, these results had broad implications. This next example is from one of the jar tests. We evaluated microsystems and toxin removal at 3 different powdered activated carbon doses using 2 different types of carbon. The wood blend performed better than the coal-based pack, and high doses were needed to achieve 90% microsystems removal. What's probably most interesting is these doses are much, much higher than dose estimates provided by published isotherm data. This is most likely due to the organic matter competition for absorption sites, as we observed in the prior study. In this example, natural dissolved organic carbon concentration in the source water were greater than 15 milligrams per liter during the bloom. So again, these high concentrations are common during severe blooms. Now, I have a 2-hour presentation on cyanotoxin treatment, but here's the real abbreviated summary. In general, conventional treatment can remove cyanobacteria cells, but is insufficient for cyanotoxin removal. Avoid pre-oxidation. Don't lyse your cells ahead of your conventional coagulation, flocculation steps. Any dose of chlorine can lyse a cell, and we found even lower doses of permanganate. Any dose above around 3 parts is really going to lyse those cells. So if you can, avoid pre-oxidation. Water systems should evaluate their cyanotoxin treatment optimization potential prior to a HAB and develop raw water triggers. So if you know your source water microsystem concentration is 5, you should know how much pack you need to add to address that. If it increases to 10, you might have to increase your powdered carbon. Cyanotoxin removal estimates by jar tests is less than removal estimated by isotherm equations. We think that's a potential impact of organic matter. Blended wood coconut pack performed well, and wood performed well in multiple studies for microsystem absorption, but jar testing is strongly recommended. Overall, as a precaution, we'd recommend that water systems have the capacity to feed up to 40 milligrams per liter of powdered activated carbon, and also test that. Some water systems have said, yeah, we can feed 40 parts, but then when they tried to do it, their system completely shut down because they had a couple U-bends and all the pack clogged their system. So we want to make sure that the feed rate is capable of being sustained, and we recommend that water systems test that out over a period of 8 to 24 hours, just to make sure that if a bloom increases in severity, they have the capacity to deal with it. AWWA cyanotox calculator is great, but it can overestimate chlorine oxidation microsystems. We generally recommend considering applying a safety factor of 2, especially if you have a higher organic matter concentrations or elevated clear well pH. Due to variability in natural organic matter, dissolved organic carbon, microsystem variants, we think public water systems should consider site-specific testing to estimate the optimal. Also, I didn't have time to go into it today, but we have found that cyanotoxins can accumulate in drinking water treatment residuals. If you are landfilling your residuals or using them as fill, that might not be a big issue, but some systems do use their residuals as agricultural liming agents or amendments, and this could have implications up the food chain and is something we are studying and we want to make people aware of. In some cases, moving on to reservoir management, it's easier to treat HABs at the source instead of within the treatment plant. Many Ohio water systems and lake managers have relied on reservoir monitoring and management as an effective treatment barrier. Strategies that have worked include algicide treatments and the use of phosphorus binding agents, primarily alum. Some additional strategies have shown promise, including hydraulic controls and ozone nanobubble treatment. And in New England, at least Kevin Sellner swears by barley straw for its allelopathic effects, but we've not really tried that approach in Ohio, so I'd reach out to him. There are a slew of other strategies that, at least in Ohio, have not worked for controlling HABs. Some Ohio water systems have spent hundreds of thousands of dollars, if not more, on various aeration and sonication approaches. In some cases, water quality was marginally improved through these approaches, but HABs persisted. Unfortunately, in at least a few cases, water quality worsened. This was the case for an expensive hypolytic aeration system in Ohio that ended up stirring up sediment, causing iron and manganese problems, and HABs actually got worse. There may be isolated cases where these approaches have worked, but I have no examples in Ohio. But thankfully, there's a bunch of emerging technologies that have worked well at the bench scale, but really need further evaluation at larger scales to determine effective and cost-efficient. Many federal and university researchers are working on evaluating advancing these emerging technologies. I did want to provide a few resources on some of the commonly used strategies. I mentioned that many states will have NPDES general permit requirements, and some even have prohibitions on use of algicide or alum. In Ohio, I imagine water systems not having algicides as an option, as this has saved many a water system from severe HABs. We do require coverage under a general permit and prohibit application to severe HABs without prior Ohio EPA approval. More information is available in the link. When I was at Ohio EPA, we received several requests to apply to severe blooms, but always tested those blooms prior to approving the use of algicides. At one water system, we detected Microsystem concentrations of over 10,000 micrograms per liter in their scums. The water system thanked us for testing and guided them not to apply algicides in this case. That next spring, I called that system and strongly recommended they start their algicide applications earlier, as soon as they began detecting cyanobacteria, prior to that bloom getting out of control. There's still a lot to learn on which algicides, at which rates, and application strategies are most effective for different types of cyanobacteria. I included a link to a new 2021 or 2020, but it's released in 2021 study on effectiveness, and work on this area is ongoing. Now, I have a 45-minute talk just on phosphorus binding agents, but this reference is a steal at $7 and provides much of what you need to know before implementing this strategy. In general, in cases where internal phosphorus loading is driving HABs, a properly dosed alum treatment can eliminate HABs for 5 to 10 years. It's great. If nutrients are still entering the system, though, more frequent treatments could be needed. Check out the book for more. And I don't get any proceeds from the sale of that book, just to be full disclosure. A newer study or strategy that OSU is partnering with the University of Florida on evaluating is ozone nanobubbles. I'm really excited about this. We'll also be testing algicide efficacy at reduced temperatures, since, as you've heard through this talk, Ohio sometimes experiences winter blooms, and algicides can lose effectiveness at those lower temperatures. We'll be conducting lab, mesocosm, and full-scale trials on Microsystem, Planktothrix, and mixed general blooms, and comparing the nanobubble treatments to algicide treatments in several paired studies. We'll also evaluate potential impacts on non-target organisms and look into the economics of each approach. Full-scale field trials are planned for streams, beaches, and small reservoirs in partnership with Ohio EPA and Ohio Department of Natural Resources. And again, we're thankful for US EPA and NOAA staff for assisting, and we hope that the lessons learned here will have broad future applicability. So stay tuned. Finally, to really address HABs, you need to address what fuels them, elevated nutrients. And even before the Toledo advisory, Ohio EPA recognized HABs were an emerging problem that should be addressed at the source. As a first step, new algae impairment criteria were developed in 2014 using cyanotoxins as indicators of impairment. The impairment listings is updated every 2 years, with the latest in 2020. Impaired watersheds will trigger development of TMDLs, or total maximum daily loads, to help address the cause of the impairment, and in this case, nutrients. We are hopeful that these new TMDLs will lead to reduced nutrient loads and fewer HABs in Ohio in the future.
Heather,
So,
I know you're at the end of your slide. And this is being recorded and it will be posted. We only have 6 minutes left until this ends. So I'm sorry if you can just give like a last minute, like a 1-sentence synopsis, and then if we can see if we can get in a couple of questions before we're done, that would be fantastic.
Sure, no problem. I know we started a little late.
Yeah, sorry.
I'll just, my summary slides up there for those who view the webinar. If you want to learn more, we do offer a 2-day HAB workshop at OSU Stone Lab, Gibraltar Island campus every summer. We demo field and lab equipment and provide in-depth presentations on the topics I covered very briefly today. The workshop's paired with an optional algae ID class the same week. I took this beautiful picture during one of the past workshops, and it's really a great location on Lake Erie and a wonderful place to learn more about HABs. So I hope to see some of you out there next summer. That's it. Thanks.
Okay, thank you all. We'd like to take some time now for questions. Like I said, there's 2 options. You can raise your hand and we'll call on you so you can ask through the voice here, or you can type in the chat and one of my staff will repeat the question. I did notice there's a couple people on phone, so we'll give you a chance after we go through the chat and ask if there's anybody on the phone that wants to ask a question, because there's no method for us to see if you have a question. So please, at this time, enter your questions into the chat for either Rebecca or Heather. Okay, I see somebody's typing in the chat. So we have a question. Is it possible to get the slide decks for the presentation? Rebecca and Heather, are you okay if we share the slide decks on our SharePoint site?
Yes, that's
Yeah.
fine.
We can share those on our SharePoint site, and if you're not already a member of our SharePoint site, you can reach out to Renee. Any other questions at this time? I figured there'd be a lot of questions. That's why Renee kind of got in, wanted to make sure we had at least 5 minutes to answer some questions. Anybody on the phone want to unmute themselves and ask a question? I see there's 2 or 3 people on the phone.
Rebecca, this is Renee Bryan. I'll ask a question real fast. In terms of all of the extensive work that you've done in your state, I think, you know, New York is, say, smaller compared to some other states in our region, say Pennsylvania or Virginia. How did this program start? Like, I'm assuming it built up to it was a smaller program, and then it kept building. And of course, with the governor's support, you received additional support for this. But are there any recommendations for those states kind of curious on emulating the program that New York has as to how they would create those building blocks to get to where you are today?
Sure, that's a great question. I think one of the most important tools is to have a way to kind of compare reports and analytical methods and that are coming from very different sources, and have a way to compare them evenly. So standardized thresholds or benchmarks to determine, what your advisory levels or what your bloom indications are, and then being a little bit flexible on those. So, some states use cell count. Can you also have a way to integrate chlorophyll measurements into that to compare them on an even playing field? It's a challenge, but that has been an important tool for us. And then utilizing these online tools like the mapping tool has been really great in order to streamline a lot of information. And then further, this initially got started with our citizen science program. So our citizen statewide lake assessment program has really been the core of our HABS program all along. So engaging committed lakeshore residents and lake associations is really a valuable tool.
Great, thank you. We do have a couple questions in the chat now. Brian asked, I saw some cyanobacteria density results presented in units of cells per milliliter. What analytical methods were used to produce results in these units? Don't most methods, especially visual methods, produce results in only colonies per milliliter?
This is Heather, and there are different ways that you can convert from colonies per mil to cell per mil. I know many phycologists will actually count individual cells, or they'll count individual cells in a colony and do averaging, or count if it's a filamentous colony, they'll count cells per filament and then measure filaments and do that. So I think cells per mil is pretty commonly used in that world. I saw the next question, I think is mine as well, and it asked if benthic cyanobacteria management strategies existed, because we had mentioned that was an emerging concern. And I can say we actually had a really kind of cool case where we had a saxitoxin, elevated saxitoxin and saxitoxin present in finished drinking water, and concentrations were increasing. We were a little worried about having to issue an advisory. No matter what the water system did, they couldn't remove it through treatment. It was really difficult. Oxidation wasn't hitting it. And they wanted to apply an algicide to their reservoir, since almost all of the saxitoxins were extracellular. So we did a whole bunch of sampling, including some qPCR work, and we found that the phanoxymenon, what was actually happening in the water column, had no gene counts. But when we did these rock scrapes along the periphery, we had really high saxitoxin gene counts. So we realized that if we had just done a regular algicide application at that lake, we would have potentially opened up the water column and increased penetration to the benthic and potentially made the problem worse. So instead, what we did, I'm sorry for this long answer, was we actually did pelleted algicide along the littoral zone around that lake, and that completely solved the problem. So we have done a lot with using weighted lines to apply algicide at depth, or to use pelleted algicide, and that has helped with some of these benthic issues. Lots of other options out there that are being evaluated.
Okay. Any more questions? We can go another minute or two here. Okay. I guess at this time, I'd like to thank you and Heather and Rebecca for joining us today. It much appreciated. It was very informative. We plan to host another webinar on our. We plan to host this webinar on our SharePoint site at some time in the future. We'll be posting it there and sharing it. Our next webinar is scheduled on November 17th, and the topic is going to be COVID, and we plan on sharing that information relatively soon. I want to thank everyone for joining us, and hopefully we'll see you at our next webinar. Thank you. Thank you again, Heather and Rebecca.
Thank you.